Okay, let's dive right into this explainer. For literally centuries, if you wanted to figure out what a plant or animal was, you had to painstakingly study its shape, its size, its colors, the whole shebang. But today, well, we are in the middle of a molecular revolution that completely bypasses all of that. We're talking about DNA barcoding, the ultimate biological identifier. Picture this. You're out in the field and you stumble across this totally unknown insect. Maybe it's damaged, or maybe it's just a tiny juvenile. In the past, figuring out what it was would take a highly trained expert, weeks of tedious study, and honestly, a fair bit of guesswork. But DNA barcoding totally flips the script. Instead of agonizing over physical traits, you just extract a tiny, standardized snippet of its genetic code. Think of it exactly like scanning a barcode on a cereal box at the grocery store. It's incredibly fast, super precise, and absolutely unmistakable. So once you've got that microscopic tag in hand, what's next? You run it through this massive, interconnected global database. This is literally where biology collides with big data. Your mystery sequence gets instantly matched against millions of validated records from all over the planet. As you can see, the whole life cycle is amazingly seamless. You go straight from catching a wasp in the wild to sequencing a tiny bit of its DNA, right to a database match. Bam! Your mystery creature is identified accurately in absolute record time. And all of this boils down to one incredibly powerful promise. One sequence, one species. We aren't wasting time sequencing billions of genetic letters here. No way. We're looking at just one specific, standardized region that completely locks down that organism's identity. But here's the catch. One single gene just doesn't work for the entire tree of life. It's kind of like needing different shaped keys for different locks. For animals, we use a roughly 650 base pair fragment of the mitochondrial COI gene. Plants, they actually need a two-gene combo called rbcL and matK. And for fungi, well, we look at a region called ITS. Fungi are actually super interesting because they present a pretty unique challenge. See, unlike animals, fungal mitochondria are full of these complex interruptions called introns. They really make a mess of things. So scientists had to pivot and use that ITS region we just mentioned, which is actually hidden deep inside the fungal cell's nuclear genome instead. So why do we use these specific genes? Well, they strike the perfect balance. The outer edges of these genes are highly conserved, meaning they're super stable. That lets us use universal primers, basically molecular handles, to easily grab onto the DNA. But the center? That part mutates rapidly, giving us the highly distinct signature we need to tell, say, a mosquito apart from a fruit fly. Getting this data actually relies on a really slick 5-step pipeline. First, you collect your sample. Second, you extract the DNA. Third, you amplify that specific barcode region using PCR. Fourth, you read the actual sequence. And finally, step 5, you blast that data straight into BOLD. That's the Barcode of Life data system. It all kicks off with sample collection, which is incredibly delicate work. A lot of the time, just a tiny little snippet of a leaf or maybe a single insect leg dropped into a vial of ethanol is all it takes. This is brilliant because we get all the DNA we need, but the physical specimen stays perfectly intact for future research. Once we have the sequence, it goes straight to BOLD, which is basically the beating heart of this whole global initiative. We're talking about millions of sequences. And it's strictly curated, too. You can't just upload junk. Every single entry requires validated taxonomy, high-quality photographs, and precise GPS coordinates. Now, when you actually query BOLD, it calculates the genetic distance between your sample and the database. And the magic number you're looking for? If you see a divergence of less than 2 to 3% in the animal COI gene, you've generally got yourself a highly confident positive species match. But listen, this entire identification system hinges on a concept we call the barcode gap. For barcoding to even work, the genetic differences between you and me, meaning members of the exact same species, have to be significantly smaller than the differences between us and a totally separate species. That measurable empty space right in the middle, that's the gap. And when you visualize this data, the boundaries are just stark. The data naturally clumps together, clearly proving that the genetic distance between two distinct species jumps up dramatically, especially when you compare it to those tiny little variations you find within a single species group. This gap is an absolute lifesaver because it solves the massive headache of cryptic species. By peering directly into the DNA, we can finally pull apart genetically distinct species that experts have mistakenly lumped together for decades just because they happen to look physically identical to the naked eye. And this tech isn't just for textbooks. It's revolutionizing the real world right now. We're using it for food authentication, like proving your expensive sushi isn't actually a cheap knockoff. We use it to track illegal wildlife trade. We are even testing river water to instantly detect invasive species, completely based on the microscopic skin cells they leave behind. How wild is that? So, step by step, we are rapidly illuminating the dark corners of Earth's biodiversity, all thanks to this ultimate microscopic tool. The next time you step outside, just remember, there is a tiny, invisible barcode written into literally every living cell around you. The only question is, what hidden worlds are we going to uncover when we finally scan them all?